Hey guys, welcome to the disease review video. A couple things that I want you kind of to think about during this time. There is not a Nearpod for this review. It's kind of up to you here. You want to make sure that if you are struggling with understanding anything, that you are using these YouTube tutorials, you're using your Quizlet, all that kind of stuff to kind of help you learn. Okay, YouTube is the bomb as far as if you want to learn stuff. And one of the best things I can do for you as a teacher is to help you learn how to teach yourself. And I know that sounds lame, but if you know how you learn and you know how you can teach yourself, it doesn't matter who your teacher is in the future, you can always learn what you need to know. Okay, so just think about that as we go through. All right. So, I tried to organize these a little bit differently this time because I know that last time trying to put in like our codons was a really a big hot mess. So I put some here and then I want to kind of show you guys how to do it. So if you just click on if you click on now it's going to be really hard with a tablet. If you click on um, if you click on the space that's blue, you can start to type. And so what you can do there is you'll be able to, like if it's GAG, then we know it's going to be C, uppercase C, and since it's DNA, it'll be T, C. Okay, and then you'll push spacebar a bunch of times, and it'll probably give you a period at first, but you can just delete that unless you can't, and then it's being a hot mess. Bum, bum, bum. So it's just kind of going to be troubleshooting with your device. So then we've got C, T, G, space, 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 etc., etc. Okay, you can go all the way down. Now this bar here, this when you look at these, when you look at the arrows buttons right there, that's where you can stretch it out. So you can pull it all the way to the end, so you can write on that whole area. Okay, now if it gets a little messed up, you're just gonna drag it. You can just drag it, drag the text box here into where it kind of makes sense for you. Okay, so hopefully with your mouse it's easier than what I'm using my finger with right now, but you can just move the text boxes around. Okay, all right. What's exactly happened in the mutated DNA sequence for the FOXP3 gene and its deletion? So if we take a look at our DNA sequences, we've got this one's good. First one's good. Yes, GAG, GAC, CGA, CAA, AAC, ATG, ACG, GAC, GAC again, ACC, TCA, AAG, GT. Okay, so in here it's TGG and in here it's TGA. Now this one is AGG and here I can see AGG and I can see CGA. CGA. Okay, so obviously we are missing one. We are missing a G here, so it's deletion. Okay, and then what happens in the cell? Basically, everything's going to shift once that one is missing. And basically, it's going to deform the whole protein. And since its job is to stop tumors from growing, since the FOXP3 gene's job is to stop tumors from growing, if it's mutated, then tumors are more likely to grow quickly. Okay, now over here I've got some replication, transcription, translation videos for you guys if you need a little refresher. I've got how to read a codon chart, I've got the circle chart, I've got the square chart, I've got types of mutations video here. So all of those are going to be helpful for this first section. Then the second section we've got is all about a pedigree. So we're going to circle to what mode of inheritance for passing it on. So it's sex linked because only males are affected. And then it's recessive because two unaffected parents have an affected child. Okay, and that's one of our key ideas for the pedigrees. Okay, and that's what I wrote down here for my 2 pieces of evidence. Now up here, I just kind of made these into long strands too, and I want to kind of show you guys how to do the text superscript. So if I do N here, and then what you want to do is up here in the home, you're going to use this button here, and you're going to do superscript. Okay, so you're just going to highlight the N's whenever you need to do a superscript, and then you're just going to go over to where it says home and then superscript to make it superscript. Okay, so however you're going to do that is just fine. Same down here for the Punnett square. We're going to use people number 3, 1, and 3, 2. So that's generation 3, 1, and 2. These are our 2 parents, and we're going to use their stuff for our pedigree. Now, this is a sex-linked pedigree, and so if you are struggling with that, I've got 2 videos about that. Here's one from Amoeba Sisters, and here's one from Khan Academy. Okay, so if you're confused about how to do a sex-linked Punnett square, we've only done a couple in class, so that's why I put those there. And then genotype frequencies, remember these are the letters. And phenotype frequencies, this is the physical traits. Okay, and then what is the percent chance of producing a child with increased risk? That's going to be 25%, because the only child that's going to be affected is this boy right here. Okay. All right, then we are going to do a little bit about the cell cycle and P53. I've put a couple of videos up about that too. I've got this P53 tumor and suppressors. I've got mitosis. I've got the cell cycle. I've got meiosis. I've got cancer and HeLa. I've got mitosis and meiosis. And then I've got why can't mules have babies? Why is cancer weird? Do we all have a cancer gene? And the answer is yes. What is it? Why do we get old and die? And how to turn cancer against itself? And why don't we all have cancer? So some super cool videos if you are struggling with some of this content to kind of help you out there, okay? So the role of P53 we know is as tumor suppressor, and it looks like it stops here during the G2 checkpoint, and depending upon your cells, it's going to tell you in the diagram what checkpoint is at. So this is part of, remember, the S, G1, and G2 phases are all part of interphase. And remember that part of interphase is DNA replication. So this is an important part where if DNA replication happens, errors can be fixed, but if not, it kind of goes through. Okay, then we have meiosis in this diagram. So it says that the capital R represents a mutation. So circle each cell that contains this mutated allele. So this cell has the R right here. This cell has the R right here. This cell has the R right here. And both of these cells have the big R. Okay, and so 50% of the offspring have a chance of getting the R mutation, because it looks like the parent is heterozygous for both. So it looks like it's big Y, big R, little r. Okay, how is this different from Henrietta Lacks? It's different because this is the process of meiosis, which is happening in sex cells. Henrietta Lacks' mutation was in her cervical cells, part of her body cells, not her sex cells, so they go through mitosis. Okay, then we have BRCA. BRCA1 is a human tumor suppressor gene found in all humans. Boop, 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 boop. BRCA1 is a protein which stands for breast cancer type 1 susceptibility protein and is responsible for repairing DNA. The BRCA gene provides instructions for making a protein that acts as a tumor suppressor. Tumor suppressors help prevent cells from growing and dividing too rapidly or in an uncontrolled way. The BRCA protein is involved in repairing damaged DNA. In the nucleus of many types of normal cells, the BRCA protein interacts with several other proteins to mend breaks in DNA. These breaks can be caused by natural and medical radiation or other environmental exposures, and they also occur when chromosomes exchange genetic material in preparation for cell division or crossing over. By helping to repair DNA, the BRCA protein plays a critical role in maintaining the stability of a cell's genetic function. So what's a tumor suppressor gene? It stops the cell cycle by helping prevent cells from growing and dividing too rapidly. This would be similar to removing the brakes from the cell cycle. Non-healthy cells would grow in an uncontrolled way and would not go through apoptosis, making more damaged cells. So this is altogether sad face bad. Okay. If a cell cannot repair the damage to its DNA, what process does the cell normally do? Apoptosis. But remember, a lot of times cancer cells don't go through apoptosis, so this is a big issue. Okay, germline or sex cell mutations of the breast cancer associated gene BRCA predispose women to breast and ovarian cancers. BRCA1 is a large protein with multiple functional parts and interacts with numerous proteins that are involved with many important biological processes and pathways. Evidence suggests that BRCA1 is involved in all phases of the cell cycle and regulates orderly events during the cell cycle. BRCA1 deficiency, or a mutation to the gene, consequently causes abnormalities in the S-phase checkpoint, the G2M checkpoint, and the spindle checkpoint, and the chromosome and the centrosome duplication. BRCA1 deficiency causes genetic instability. BRCA1 mutation triggers responses to DNA damage that blocks cells from dividing and induces the cells to undergo apoptosis. The BRCA1 mutant cells cannot develop further into full-grown tumors unless further mutations occur. However, if further mutations occur to the genes that control contact inhibition and the apoptosis mechanism, cancerous tumors can develop. So what is meant by germline mutation? Germline mutation are mutations that pass on to offspring through meiosis. Okay, how does the normal BRCA1 gene play a role in homeostasis? So remember, homeostasis is that fancy word for balance. It's involved in all phases of the cell cycle and regulates cell cycle progression. So if this is like a circle or cycle of life, the BRCA1 gene tells how fast it goes through these cycles. How does the BRCA1 gene disrupt homeostasis? So if it's mutated, then it triggers responses to DNA damage that blocks cells from dividing and induces cells to go through apoptosis. So this gene helps signal to a cell, hey, you're damaged, you need to die. What else is required here? So it says further mutations occur to the genes that control contact inhibition and the apoptosis mechanism, and cancerous tumors can develop. Okay, and so if a tumor is formed, then we know that the rate of cell growth increases and we have uncontrolled cell division. So what's contact inhibition? What's apoptosis? What's tumor suppression? Hopefully you guys remember those. And then which of the checkpoints is interrupted by the BRCA1 mutation? It looks like all of them, essentially. So the BRCA1 mutation is super important for all of the checkpoints. All right, then this last part here, we made this in your paper notebooks back in the day. So if you want to do it on that, that's great. If you want to do it in here, that's also great. My suggestion would be that you use some words and pictures in here, even if you just use pictures from the internet, because these are our 5 big questions from the whole unit. All right, so once you're done with that, you're all set, ready to take the test. Good luck.